Okay. So we've started our live stream on YouTube as well. In case you see that there's anybody who you know is trying to get in but cannot, you can also find us on our YouTube channel. Um, it's 6:01, so we don't want to be late for this one. We have Dr. Devinsky with us already, and we have an hour for the webinar. So I just want to give a quick introduction before we begin about Gateway School and Dr. Devinsky. For those who don't know, the Gateway School of Mumbai is a lab school dedicated to empowering the lives of students with learning challenges. Together with our students, their families, and our community of teachers, professionals, and support staff, we work towards our common vision that students will lead fulfilling, productive lives and be active contributors to society. Apart from working closely with students to fulfill our vision, we also work with the community at large so as to spread the best practices we are seeing at Gateway. We have a 6-month in-service teacher training program that has gone virtual this year. We've had people attend from all over India and even internationally. Our R&D department has launched India's first ever app to monitor and build foundational literacy skills. We're currently piloting this with over 500 students. We've been recognized for the work we do. We've been an Ashoka Changemaker School since 2016. In 2019, we were recognized by Great Place to Work as one of the 10 best NGOs to work for in India. Over the next few years, we hope to continue our initiative to fulfill our vision. We plan to open an intervention center that will serve a wider range of students with disabilities, as well as adults with disabilities. We also plan to launch a certificate course that will help young adults with disabilities gain employment opportunities. That's a very quick background about Gateway School of Mumbai. Moving on to introducing our guest speaker today, Dr. Orrin Devinsky. Dr. Devinsky is a member of Gateway's advisory board. He is a professor of neurology, neuroscience, neurosurgery, and psychiatry at the NYU School of Medicine, where he directs the Epilepsy Center. His epilepsy research includes autism, genetic epilepsies and their treatment, healthful behavioral changes, quality of life, and surgical therapy. Other interests include behavioral neurology, evolutionary biology, nutrition, and history of science. He serves on the boards of the molecular biology companies Tevard Biosciences, Regal Biosciences, and Script Biosciences. I don't want to take up more time, so I'm going to hand it over to Dr. Devinsky, before which I just want to remind everyone, please have yourselves on mute so that there are no disturbances for the speakers. As well as if there are any questions, please feel free to post them on chat and we will sift through them. And in the last 15 minutes, we'll throw it open for a question and answer session with Dr. Devinsky. So Dr. Devinsky, if you're ready, we're ready.
I am. Thank you very much, Miriam, and thank you all for this very kind invitation. I look forward to sharing with you some of my thoughts on an overview of epilepsy and autism with some interest in molecular biology, diagnosis, and some new treatments that have come out. So to begin with, there are many challenges in looking at the relationship between autism and epilepsy. There are many large groups that have looked at autism patients from the perspective of genes, from clinical features. But in many of those cases, when epilepsy is evaluated, it's a checkbox. Does the child have a history of seizures or epilepsy or not? But it's not an in-depth one. So often those studies, some of the best ones done, for example, don't look, is it focal or generalized epilepsies, which is like saying, is it a man or a woman? Those are the two broad categories. And then some of them have limited follow-up. So they assess a child at age 8, but that child may develop epilepsy at age 12, and that child will be listed as someone without epilepsy when indeed it turns out they do have epilepsy. And then there are different forms of epilepsy, as everybody knows. Some individuals with autism may have 1 or 2 seizures and live the rest of their lives without another one, where some individuals with autism may have seizures every week despite taking 3 medications at high doses, which cause significant side effects. So the spectrum of epilepsy severity within people with autism varies very widely. And then there's the role of epilepsy as an endophenotype. Is there a way to divide autism patients? And there probably is, And the presence of epilepsy may separate a group. We know, for example, many of the genes that are known to cause epilepsy and autism will be highly concentrated in this group, as an example. So in looking at autism spectrum disorder genetics, as I mentioned, many of the large studies, and this is from a few years ago, but these are published in Nature, Cell, Neuron. These are, 3 of the top 5 world's leading scientific journals, and they find many examples of mutations, which is what a lot of these genetic studies are looking at. But you see some of them involve literally thousands of individuals with autism. But there's no mention of epilepsy in any of these studies, for example. So these are studies published in huge journals looking at individuals with autism, but they never even ask what percent of the patients, if any, had epilepsy, and does that segregate some of the genetic findings, which it probably does. And then what about the opposite question? So as I'll get to, there's no doubt that we know epilepsy is more common in individuals with autism than in the general population. By age 20 or 30, up to 30% of people with autism will manifest at least one seizure, and even more will have epilepsy waves on their EEG. But if you have a child with early onset epilepsy, do they have an increased risk of autism? And the answer is yes. So any severe epilepsy beginning in the first 3 years of life, infantile spasms being probably the most severe and the most early, is associated with very high rates of autism spectrum disorder and intellectual disability. So below you can see some of those bullet points. If epilepsy begins before age 2 years, 10 to 14% of those cohorts of children will be diagnosed on the autistic spectrum. If they have infantile spasms, that number is probably closer to the upper 30%, between 30 and 46%. By contrast, if epilepsy begins after age 2 years, the frequency of autism is 5%. So again, I think one of the messages about autism is that anything that perturbs brain function, either in utero or in the first 2 years of that child's life, has a very high chance of causing autism by disrupting the normal brain development, whether that dysfunction be literally a stroke in the mother's womb, whether it be a genetic abnormality that manifests at age 6 months of development during brain development, or something that occurs at age 1 year, for example, the onset of bad epilepsy. Very diverse genes, very diverse environmental factors, and combinations of the two can produce autism. So as I said, what's the rate of epilepsy features in individuals with autism spectrum disorder? And by age 25, epileptiform activity, depending on the study, ranges all over the place, but it's pretty high. So epileptiform activity, my metaphor is these are spikes or sharp waves that are seen on EEG. They are not seizures. So my metaphor for epileptiform activity is that clouds are to rainstorms as epileptiform activity is to seizures. So anyone, if there are 100 people on this call and one person had epileptiform activity on their EEG, which might be the average in a population, number one, there's a 50-70% chance that person already has a known history of epilepsy. And if not, then there's a 20% chance they will develop epilepsy in the next 5 years. But as far as the actual diagnosis of a seizure or epilepsy, it ranges from 20 to 40% by early adulthood. It's interestingly a little bit more even sex ratio. So it's very well known that individuals with autism as a broad group have about a 3-to-1 male to female ratio. But for those with epilepsy, it's not 1-to-1, but it's more like 2-to-1. And in the more severe epilepsy cases, the ratio of girls to boys actually gets pretty close. It's often been reported that there are 2 peaks for the onset of epilepsy among individuals with autism: early childhood, which would be cases which I would refer to as the more symptomatic forms of autism, where we know that there's a significant genetic abnormality or it's part of a syndrome associated with other physical or neurological features. And then one in adolescence, and these are children for whom standard genetic screens are pretty negative, and there are no dysmorphic features in the face and nothing else to suspect more of a syndrome beyond autism itself. But again, these are far from pure, and there's a lot of overlap on both sides. So this is a study that looked at the prevalence and development of epilepsy in an autism population. And on the left side is from this study that was published by Devinsky et al. And on the right is a database of 2 epilepsy centers I'm involved with. And what you can see is that after age 13, the prevalence in the first study gets up to 26%. And there's a suggestion of 2 peaks, but it's not really so clear that those peaks in larger data sets are there. Certainly, we do see on the right side, I think I'll use my mouse here, but you can see over here, there's a pretty big rate of the development of epilepsy in the first 6-7 years of life. It never disappears here. So if a child is age 11 or 12 or 14, there's still a pretty high number. In our group, age 15 was high, but then afterwards it tapers down. And of course, like with adults in the general population, it never goes away. And for people with autism, it's going to be higher at age 27 or 24 still than the general population for new onset epilepsy, but it's not that common after age 20. It still happens, but certainly most of the development of epilepsy will occur by age 20 in individuals with autism. Well, what can we say about epilepsy in individuals with autism? And I would say it's a marker or a contributor, and probably both. And a marker means it's just something that tells you something, and then a contributor is it also leads to disease. And just to take an aside for a second, if you look at heart disease, many people would say, for example, that very high cholesterol over 250 or 300, wouldn't be much controversy, certainly is a biomarker for increased risk of heart disease, especially in males over 50. And it's also a contributor. We think that that's not just a marker, it actually is part of the disease process. But one can also say that gray hair is a marker for heart disease in men, because men age 70 are going to have high rates of gray hair, whereas men age 20 are going to have low rates of gray hair. So gray hair is a biomarker for risk to heart disease, but by no means is it a contributing factor. So you got to separate them, and they're not always so clear. So one thing we know is the lower the IQ, the higher the risk of epilepsy, and this has been borne out by essentially all studies. And the same is true for other skills. The lower the social skills, behavioral skills, motor skills, activities of daily life, the lower the functioning, the more likely that individual with autism will develop epilepsy. So the individuals with Asperger's who are very high functioning, who may have IQs in the normal range, are going to have, they're going to have increased rates of epilepsy, but they're going to be much lower than a nonverbal child with autism, for example. And among those who get epilepsy and have a diagnosis pre-existing of autism, epilepsy will remit in 15% and will be controlled in around 45%. So, the prognosis is pretty good for the vast majority of patients. However, it is treatment resistant in a significant portion of those with both symptomatic as well as idiopathic autism. And idiopathic means there's no obvious cause that's been identified on an MRI or gene panel. And again, what predicts treatment-resistant epilepsy? And this is essentially a definition from the International League Against Epilepsy, where if a child or an adult is treated with 2 appropriately chosen medications, that means the medicine's right for the epilepsy syndrome that child has, and they get to therapeutic doses or blood levels of 2 drugs, and they continue to have seizures, that's treatment resistant. So early onset of epilepsy, developmental regression, significant motor and language delays all predict a more difficult time to control those child's epilepsy. And as is true for people without autism, focal epilepsy beginning in one or several different regions of the brain is more treatment resistant than epilepsy beginning everywhere in the brain, which is referred to as generalized epilepsy. And the same rough relationship holds true for individuals without autism. Focal epilepsy tends to have a much higher rate of treatment resistance. So epilepsy was long considered a pretty benign disorder, but I think we've changed that now. We know, as I think I'll get to, epilepsy, unfortunately, is probably the leading cause of death. Epilepsy is likely the leading cause of death among individuals with autism. but on a much more subtle basis, it probably contributes to cognitive and behavioral disorders. On the behavioral disorders, which are well established to be issues among people with epilepsy, mood and personality disorders, anxiety, psychosis, and cognitive problems extend through the full range: attention, memory, executive functions, and language. The effects at times can be destructive and progressive, typically very slowly. So we know from many studies that individuals with epilepsy will have smaller brain volumes than those without epilepsy. Even if it's someone like you and I who had a normal IQ and normal functioning and then has epilepsy and continues to have epilepsy for 5 or 10 years, typically when you do quantitative brain imaging, you can show that after 5 or 10 years of epilepsy, the brain can change. Now, no one can 100% say who's the chicken and who's the egg, but we think in some cases the seizures themselves contribute. And in other cases, the actual epilepsy waves contribute. So again, I said epilepsy waves are like clouds. And yes, if it's cloudy out, you don't need a raincoat, you don't need an umbrella. It may tell you you're in a place where it rains at some time, but the clouds can affect the amount of sunlight you get. It might affect your vitamin D levels in a place like Northeast United States, where sunlight is limited to 4 or 5 months a year, which can make vitamin D. So there's one pattern, which is shown here on this slide, called continuous spike wave discharge of slow wave sleep. And this occurs, in a small population of people with autism in childhood. And we think in some cases, this really can drive a lot of the cognitive and behavioral disorders. And in some cases, and I've seen this, a child who's nonverbal, who's got terrible behavior and terrible attention, has this pattern identified, has it treated with something like IV steroids, which is a serious treatment. The EEG pattern gets dramatically better and the child starts speaking. It literally can be a miracle. Now, that's not common. I don't want to give false hope that this is something that's likely going to treat a large group of people with autism, but it does exist. And when it's found, it sometimes can suggest very specific therapies. In addition, this pattern tends to abate over time. So if we were to take a place like NYU and look at all our admissions for pediatric epilepsy, nothing to do with autism, just everybody with epilepsy going to the child unit, it's probably 5 or 7% of all admissions. And if you go to the adult unit over 18, it's less than 1%. And the reason for that is that most of these children with this pattern, it will be outgrown somewhere over the course of childhood. We don't know why and what's happening, but it does happen. So this is a slide I find incredibly informative. This is from an old book written in 1979 by Manuel Gomez, who was at the Mayo Clinic at the time and was America's leading expert in tuberous sclerosis complex. This is a genetic disorder that has a high rate of causing both autism and epilepsy. And this is a table from his book, and he just looks. If epilepsy begins before age 1, 7 out of 79 children had normal intelligence. So essentially 10% have normal intellectual function. Like the word mentally retarded, it should be intellectually disabled, but 1979, that was a commonly used word. And by contrast, if epilepsy begins after age 15, only 2 out of 13 individuals had intellectual disability. So essentially the opposite ratio, about 15%. And of the 19 children in his cohort who never developed epilepsy, it's very common in tuberous sclerosis, none of them had intellectual disability. So just to pause here for a second, keep in mind, those with tuberous sclerosis who are going to get epilepsy early in life often have a more severe form of it, often have a more severe gene. There's 2 genes that cause it. The TSC2 gene tends to cause a more severe disorder than the TSC1 gene. They often have a higher tuber burden. There are these benign tumors in the brains of these kids. And so those with early onset epilepsy tend to have a higher disease burden on the MRI. So you can't just say it's all from epilepsy, but I will tell you a significant portion almost certainly is from early life epilepsy, and that's supported by the data from everybody with infantile spasms. So most of these kids who develop epilepsy in the first year of life with tuberous sclerosis did have infantile spasms. So the point is, epilepsy is bad for the brain, and it's worse for the brain if you're a very young child under age 2 years. I will add just one hopeful note here, and this is a patient I saw now, I think 24 years ago. It was right after vigabatrin came into the United States, or we were able to get it from Europe. And vigabatrin is a very effective drug, specifically for children with tuberous sclerosis and infantile spasms. Anyway, a patient got admitted. I actually knew the family through some people, saw them pretty quickly. Child had infantile spasms, had an MRI done. The MRI showed tubers. It was pretty clear that this child had tuberous sclerosis. We started the child on vigabatrin within literally hours. The seizures came under control. That child graduated from a fantastic university and is extraordinarily bright. Had he have been born in Dr. Gomez's time, where vigabatrin wasn't available and they would have given him phenobarbital and dilantin, 90% he would have been intellectually disabled and would never have made it to a community college, never mind a fine university, and graduating and doing well. So what are some of these lessons from tuberous sclerosis? And I think it's informative. This is a picture of the MRI showing these tubers that are outlined in the red lines, those white areas that should not be there. And what are the risk factors for autism in individuals with TSC? Well, early age of seizure onset, frequent seizures, seizures beginning in the left temporal lobe, which is a very important area for language and language comprehension, and a high tuber burden, that is a large number of tubers in the brain. And you can see the tuber burden on the slide below from an article published about a decade ago. And those with autism, for example, in this slide, have a much higher tuber burden than those without autism. So again, the severity of the neurologic insult predicts the likelihood of autism across any cohort. This is just one specific disease that's been well studied, but I think this is essentially true for everything that's been examined. So this is my own slide, and I think it's helpful to think about as we go to think about drug therapy for individuals with autism, whether it's a psychiatric drug, a neurologic drug. But if you look, this is kind of a bell curve of forms with a little bit of a skew on the upper end, but adults are in the middle, and that's just the general population, people who are on this call right now, myself. You know, I can predict how most people will do. If I give you penicillin, yes, 7% will get a rash. A few people may get a diarrhea, but you know, we have a good sense of how it's going to work. If I give you Tylenol, Advil, if I gave you a stimulant medication like methylphenidate, you guys, your heart rate would go up. You'd be kind of focused. You'd be a little nervous. But when we give it to kids, which should be the middle red line here, which I'm outlining with the cursor here, they have a wider range. So yeah, some kids, when you give them methylphenidate, a stimulant for ADD, they actually get wired like adults. Many will get calmed down, which is the beneficial effect in attention deficit disorder. But on the widest line above, when you treat children with autism, I think their response to medications becomes even less predictable. They have a wider variation of how they respond. Number one, there are 200 different genes and many environmental factors that cause autism. So their neurologic phenotype, so to speak, is really partly determined by those very different genes. And so they'll manifest, but how they respond to medications is more variable than it is in regular children or in adults. So this is just a slide that summarizes, a tiny fraction of the genes that we know that are shared across epilepsy, autism, and schizophrenia. And again, you look at tuberous sclerosis, Dravet syndrome, DUP15Q, Rett, and then a number of other genes, catnip2, and then gene duplications or deletions, parts of the chromosome, which there's an extra copy or they're missing a copy. Those are all going to be cases where you can get essentially all of them have epilepsy, autism, intellectual disability. Some also have cortical dysplasia, an MRI abnormality, similar to tubers, and some will also have schizophrenia. So some of these genes cut across many different neurologic and psychiatric disorders, and how they manifest depends in large measure on the other 20,000 genes that the child has. So just because I have a tuberous sclerosis gene and my daughter has a tuberous sclerosis gene, I may not have any symptoms. because some of my other genes may protect me, but she may have gotten a gene from my wife that doesn't protect her, and she may be severely affected by that disorder. So genes are answers, but they're not in any way the complete picture. So before we turn to some therapies, just some thoughts on the convergence of the etiologies of autism, epilepsy, and intellectual disability. You know, for brain disorders, it can be a single protein and gene that's affected. It can be a metabolic pathway in which many genes play into it. For example, I mentioned tuberous sclerosis. That's the mTOR pathway that leads to cell growth in the brain and in other tissues. It's an important pathway in cancer as well. And it's also a gene pathway, not the tuberous sclerosis genes themselves, but probably 5 other genes that affect this pathway that can also give rise to epilepsy, autism, and brain developmental abnormalities. And then there's just the effect of influences, genetic or otherwise, on the development of the brain. The brain is already forming 6 to 8 weeks of gestational age. The nervous system is forming, and it continues to be forming, you know, all the way through adolescence. So there's a large time, but it's that window of in the mother's womb in the first few years of life that's really critical. There's genes that are how different nerve cells connect at the synaptic junction, which are very important. And then there's just the overall network, how brain cells synchronize. Any disruption at any of these levels can contribute to all of these disorders, autism, epilepsy, and cognitive impairment. And again, the earlier the age of the effect and the greater the severity, the more likely these 3 disorders will all occur, and the more likely they'll be severe. Going forward, I think it will be really important to get more crosstalk, and this is happening. I think science has gone from my center did this and your center did that to we need to collaborate across 10, 20, 50 centers to get the kind of numbers and statistical power that we can really start to sift through the potential multitude of causes and really identify, you know, what's a confound and what's a cause. And that's complicated in science. So I'm going to turn now for a little bit, because I know cannabis has emerged as an interesting therapy in both the epilepsy and autism communities, and talk about its use and some of its background. So we'll start out with the biology. There are 2 main species of cannabis, sativa, which was described in China 12,000 years ago, indica, which comes from your home country, described in Sanskrit over 3,000 years ago. In general, the higher the ratio of THC to CBD, the more psychoactive it is. So THC is the major psychoactive ingredient in cannabis. It's why people, young individuals or older, use it to get a high from it. Whereas CBD is in no way psychoactive. And they're both biologically active compounds. In America, people often talk about sativa as being high in THC and indica being high in CBD. And that was true 1,000 or 500 years ago. But certainly in the last 50 years in America, and I have no idea what the purity is, plants still in India, but they've been very, very highly interbred. So there is no such thing as sativa and indica in the United States, but it may well be in India and China. I'm not an expert on that. So interestingly, cannabis, the plants themselves have over 500 compounds. It's enormously diverse. And cannabis isn't especially unusual in this regard. Many other plants have many, many, many compounds, and they're all there to support the plant's biology and survival and reproduction. That's why they're there. So as I said, THC is the major psychoactive ingredient. We know exactly how this works. This is the endocannabinoid system that was described back in the 60s and 70s. But essentially, there are 2 cannabinoid receptors, CB1, which is mainly in the brain, the central nervous system. That gives rise to the psychoactivity. The CB2 receptor is mainly outside the brain in the immune system, although there's a little bit of it in the brain, and that's, you know, not psychoactive. Cannabidiol, as I said, is a compound I've been very involved with studying for epilepsy. It is not psychoactive. There's no tolerance. There's no dependence. People can take it for a month and stop it, not go through a withdrawal. And it works on many receptors. I listed a few here, but the GPR55 receptor is where we think it may be its most important anti-seizure effect. The TRPV receptors are involved with pain perception and many other physiologic processes. By the way, hot peppers, capsaicin stimulates one of the TRPV receptors. And then serotonin receptors, we think that may be part of the reason why CBD can be helpful in treating anxiety in some people. So these are some of the compounds showing their structure. THC on the top, CBD and another drug that's being evaluated. But I would just point out, and there's no reason, I'm happy to share these slides so you can get them, but there's these compounds are complicated. I simplified THC in saying it works mainly on the CB1 receptor as a stimulant of that receptor, but it does a lot of other things too. Same with CBD, and the list is by no means full on this slide. So these compounds do a lot of different things in our nervous system. So what about the evolution of cannabis? I believe Devinsky's quote from 50 years ago that nothing in biology makes sense except in the light of evolution. So the cannabis family evolved about 15 million years ago. Hops was a branch off of that. Hops is in many beers and evolved 6.5 million years ago. And cannabis actually smells a bit like hops, and that's because they share many compounds. So as I said, why are there 540 compounds? Well, there are not 540 compounds to help human beings. This plant's been evolving for 15 million years. Humans probably only started using the plant regularly in the last 10 to 20,000 years. So the plant's compounds are there, nothing to do with human medicine. And they're there for the plant. It takes the plant energy and time to synthesize cannabinoids, terpenes, flavonoids, the major compounds that it has. And it does it for the reasons that I said, to help itself, to combat viruses, bacteria, funguses, insects, herbivores, to prevent desiccation, to help with reproduction. And that's why these compounds are there. And where was it first domesticated? Well, probably in Central Asia, probably in China. And initially we have from the historical records, it was used to make fiber, rope and cord, used in religious ceremonies, used medicinally, and then probably much later used recreationally. And how quickly does artificial selection work? It works quickly. And I just show here, the changes to this breed of dogs. The one on the left is from the late 1800s. The one on the right is from about 100 years later. But many aspects of their physiology, the length of the legs, the size of the nose and snout, have all been selected for. So I eat a lemon and there's very thin rind and there's a lot of fruit. I have a print from a lemon from 200 years ago. The rind is about 5 times thicker and the fruit is much more fibrous. And here's a picture of corn. Teosinte is on the far left. The amount of starch in a teosinte is probably equivalent to about 3 or 4 kernels of corn, and there are probably about 100 kernels of corn. So, the amount of starch in a modern corn plant is, 30, 40-fold higher than the wild ancestor. So I may eat corn and others may eat corn and say, well, I'm eating a natural food. It comes from the ground. It's an organic farmer. It is a natural food and it comes from the ground. And if there's no pesticides, et cetera, that's great. The only problem is it is genetically modified. It was not done with CRISPR or some fancy gene therapy, but it was done by human beings over generations and generations who changed it radically. No one 10,000 years ago who was eating teosinte would have any idea what corn was. It would have no bearing. It would be like taking a Neanderthal without clothing and putting him on, a bus in Mumbai. He would stand out quite dramatically from the rest of the population. Things have changed, and not that many, their genes are quite similar to ours, but you change some specific ones as they did in corn and things change dramatically. So we're also not terribly natural. So in the last 12,000 years, what's happened to human beings? We have thinner skulls, we have smaller jaws. For example, I'm not sure what the rate is in India, but in modern United States adolescents, I would say 80% have some wisdom teeth removed. And that's not natural. You go to skulls 500 years ago, there was no problem with impacted wisdom teeth. It didn't exist. That's a 500-year-old problem. And it's pretty clear, one of my friends and colleagues, Dan Lieberman at Harvard, who's the head of human evolutionary biology there, I think he's exactly right. He says this is because when children are given food, their food is all soft and processed. It's mashed fruits, it's mashed grains. They don't chew. Human beings are meant to chew their food, but we've processed our food and we've selected food that's softer and softer that we no longer do what we're supposed to, so our jaw doesn't grow to the right size. And then when we turn 16 and our wisdom teeth come in, the jaw isn't the right size for those teeth. Milk tolerance. Mammals evolved over 200, 215 million years ago. Humans are the first species to consume milk after weaning from the mother's breast. And when did that happen? It happened about 5,000 years ago, and the genetic mutations occurred in at least 4 separate populations affecting 4 different parts of the lactase gene. And that's because humans started becoming herders of sheep, goats, and cows. And then it became clear that those who could tolerate drinking milk at age 5 or 15 or 35 would do better, because when water supplies were low or were contaminated, when nutritional sources otherwise were not available, those who had this gene, and that gene spread through populations quickly in a matter of generations. And then there are genetic adaptations to infectious disease. Many of the severe diseases that affect Western societies are thought to be adaptations to cholera, to malaria in the past, certainly in the Mediterranean region and Africa, and cystic fibrosis. So many of the diseases we may now have represent genetic adaptations that our ancestors had to combat infectious diseases. And certainly most of the diseases that will kill the people on the call today, obesity, diabetes, cancer, dementia, hypertension, heart disease, to the much more mild ones like constipation and back pain, they're all mismatched diseases. These are things that if you go to an indigenous population 150 years ago, who may have been a farming community, may have been a mix of hunting, gathering, and farming, none of these existed. Basically none of these. The rate of cancer among Native Americans in 1900 was about 1/100th of the rate it is in Americans today. And people say eating meat is terrible. Those people were eating bison for 70% of their calories and had a 1% cancer rate, had no diabetes, had no obesity, had no hypertension, had almost no dementia. So, and I'm not saying meat is good. Plants can obviously be very, very healthy, but refined carbohydrates, sugar, white rice, white flour, I would suspect are the leading causes of death around the world. So one of the other things with cannabis that I faced in my medical population is that people love it because it's natural. The fact that, some big drug company in New Jersey is manufacturing some compound like topiramate, that's a drug, and parents don't like giving drugs to their kids. But a plant is very appealing because Mother Nature, is pretty nice, and she's done well by us historically. But of course, Mother Nature's not always so sweet and lovely. This is a puffer fish, or as the Japanese call it, fugu. Fugu makes a toxin called tetrodotoxin, TTX, and 1 milligram of it, a tiny, tiny amount, will kill you in probably 30 seconds. So it's very natural. Poison ivy is natural. Sharks are natural. Many things are natural and they will kill you. Obviously, most things that are natural don't kill you, but mushrooms are wonderful foods, and they can also be terribly dangerous foods. So I think we often tend to worry about the wrong things. As I was hinting before, genetically modified organisms are highly feared in America. But India is a country, when the population bomb was written in 1962, it predicted there would be mass starvations in the order of tens of millions, if not hundreds of millions in India over the course of the next decade. And thanks to several genetically modified organisms, that never came to pass. And indeed, India became a net exporter of food due to those genetically modified crops that produced more starch, that resisted bacteria and other pests without requiring pesticides. So it saved millions of tons of pesticides. Again, I'm not Mr. GMO, but I think when you look at the cost benefit, GMOs have saved millions of lives and prevented millions of pounds of pollution in every country. And this brings me to something that is relevant to several points. In America, there's a very big belief in this entourage effect that, CBD doesn't work without THC. You need both of these together. And this entourage effect was actually named by Ben Shabbat and Raphael Mechoulam, the discoverer of THC and CBD in Israel. But it was about a totally different thing. It was about a lipid 2-AG, which is in your brain, and it stimulates the CB1 receptor. It's the natural stimulant for that receptor. THC, of course, is not something our bodies make. It's something we consume from cannabis. But there are some esters in the brain that also exist of 2-AG derivatives. They don't bind to the CB1 receptor, but when they're present with 2-AG, they greatly magnify the effect. And they refer to this as an entourage effect. That is, in the presence of the natural stimulant, that stimulant is much more effective. So people will take that and say that CBD and THC act synergistically. They may. There's just no data. No one studied it. There's no experimental anecdotal data from in animals, and the human data is really parents basically saying, I gave it to my kid and they were cured. And no offense to parents, but scientists are wrong all the time. And when parents talking about their kid, placebo response rates are twice as high. So I'm just quoting from one terrible website. New research confirms what has long been thought. CBD, in addition to other entourage cannabinoids like THC, is far superior medically than pure CBD. And this is just wishful thinking. It may be true, it may be false. No one studied it, so no one knows. So I'm going to turn now to briefly CBD to treat epilepsy and then turn to autism. As I said, epilepsy occurs in probably greater than 25% of all people with autism by adulthood. And we know from some studies I've done with many colleagues around the world that CBD does reduce seizures in Dravet and Lennox-Gastaut syndromes. And this has been established in large, well-controlled trials published in major journals. However, in 2 other trials, it had no benefit for focal epilepsy and has not yet been studied adequately in generalized epilepsy. So we know CBD works for some rare epilepsies. We don't know it works for the vast majority of people with epilepsy. Dravet and Lennox-Gastaut syndrome are well less than 5% of all epilepsy. What about medical marijuana, cannabis for autism? In the United States, most people can get medically approved cannabis. The products vary in what they have. Their quality control is limited. And the consistency, if you buy something in New Jersey in March, if you come back in June to get another supply, it may be even a different strain of cannabis, and certainly the conditions under which it's grown may have changed as well. And we just really lack safety and efficacy, how well it works data on THC in autism in any way. And we really need controlled trials. There's a lot of religion. For me, religion is belief without any evidence, and science is doubt despite compelling data to support a hypothesis. So as a scientist, you always doubt, and if you go to the leader of a religion or a deep believer, they believe and they don't need any data, and that's okay. I'm not against religion, but it's just important to separate it from science. So there is a little bit of data on CBD and THC in autism. There are some mouse studies that have found some positive findings on behavior. There's been some clinical styles. Zynerba is a company that uses a synthetic CBD given by a skin patch, and they've studied it in Fragile X, but they did find improvements in social avoidance, temper tantrums, repetitive behaviors, and hyperactivity. There's mountains of anecdotal data. I've treated hundreds of people with autism with CBD, and I do think as a clinician, many parents who I trust have felt that it has improved anxiety, has improved sleep, has reduced disruptive behaviors. But the clinical studies to support that scientifically are still in progress. I think some of them are ongoing, and we should get data hopefully in the coming years. So if you look, for example, on just the question of what's out there and what's the belief system, if you look in the National Library of Medicine, this slide is from a few years ago, but autism and CBD, there were 2 papers. Autism and THC, there were 2. Just the word autism, over 41,000 articles. But the ratio of articles on autism to autism plus CBD and THC was 10,000 to 1. If you go to Google, and again, this is 2 years ago, these numbers are going to be much larger. The number of links on autism and CBD was over 400,000. Autism and THC, over nearly 400,000. The number of links on autism overall was 121 million, but the ratio was 150 to 1 on the public sites. In the scientific sites, it was 10,000 to 1. So again, the point being, the scientific data is lagging way behind the popular belief system. And so just to finish up, what's the role of scientific medicine? Well, we think of science, and the slides on the left, top left, as a clear, clean road. Everything's nice. There are scientists in white coats, which sophisticated equipment, doing studies, and from there we glean truth. But the reality is science is a dirty road. On the top right, you see a terribly littered roadway with a couple car wheels on the left. But that's what science is. You know, if you look at the bottom, there's a leech with blood on the hand of someone. And in the 1800s, bleeding, whether it be by lancet or leeches, was one of the most popular medical therapies throughout Europe, America, and many other countries. It doesn't mean it was right. It probably didn't work for 99% of patients treated, and even worse, it probably hurt and sometimes killed many of the people who were treated by doctors from major universities across Europe and the United States who are trying to do the best things for their patients. And then when you get to autism, certainly where I practice in New York, chelation, secretin, Lupton, hyperbaric oxygen, gluten-free, casein-free, stem cells, antifungals, camel's milk, nicotine, you name it, dolphin therapy. There are believers in all these therapies, but there's no data for any of them. And you can just go through an infinite number of rabbit holes. And as one Nobel laureate said, for every scientific hypothesis that's proven true, there are an infinite number that are false. And I think I think that's a very wise word. So in the future, for studying cannabis and its role in autism, I think we need scientific data, and we need it soon. We need to assess things individually and in combination, and they may differ for different people. Certainly what works for some children, we all know will not work for all children. And some children may get benefit and some will get terrible side effects. So we need to just be open-minded. And my religion is science. I believe in doubt. There's one saying, I believe in God, everyone else show me the data. So I will finish on that note, and thank you all so much for this kind invitation.
Thank you so much, Dr. Devinsky. This was a very, very informative, very enlightening session. Even though it was 45 minutes, we were completely riveted. I'm not going to take up too much time. We're going to quickly move on to questions that we've been having ever since you started talking. So let's have Nikisha on, and she can sift through the questions. Nikisha, are you there?
Yes, I'm here.
Lovely.
Dr. Devinsky, we have a few questions that have come in. The first one is there a link between a febrile seizure in a newborn and them developing autistic spectrum features?
So it's a very good question. I should say that at the outset, it's very unusual to have febrile seizures in a newborn. So febrile seizures, depending on the definition, it's typically 6 months to 6 years of age is where febrile seizures are diagnosed. There are some groups that lower that to 3 months of age or even 1 month of age. But so a newborn, certainly in the first weeks of life, would never be called a febrile seizure. Maybe at age 1 month or 3 months would be the earliest. Overall, the relationship between febrile seizures and autism is extremely low. So febrile seizures are not considered to be a risk factor for autism overall. Obviously, it will happen to some by coincidence, and possibly, extraordinarily rarely, there may be a true relationship. But if it's there, it's not been borne out in large population studies.
Can I move to the next question, Doctor?
Please, yeah, please.
Is 5 years with no seizures for a child on the spectrum enough time to start weaning him off his epilepsy medication?
I think it's a reasonable thing to consider. My only caveat there would be that I think it's important to check EEGs. I prefer at least like a home EEG for 24 hours. And certainly if there are no epilepsy waves after 3 years, 4 years, 5 years, and the child's been seizure free, yeah, I would start to wean medications, but just do it slowly. If the EEG does have epilepsy wave activity, then I would be a bit more cautious, and the risk of a seizure recurring is much higher. You can ask the next question.
Doctor, something that connects to the previous question, is it safe to assume that the activity is only going on and it's not manifesting?
So I think you mean as far as the epilepsy wave, I'm not sure what the questioner wants, but it's hard, clearly with epilepsy, there are seizures which we can see, seizures which we don't know about. They happen at 3 a.m. and they're not severe, or the parents just are sleeping and don't know that their child in the next room is having a seizure. So there can be big seizures that nobody knows about. And then there can be subclinical seizures where we look on the EEG and it looks, smells, and tastes like a seizure, but it turns out, the husband was with the person or the mother was with the son at that time playing a game, and they said, no, I was with him exactly when that time, and he was fine. And so we'd call that a subclinical seizure. So there's everything from epilepsy waves to subclinical seizures to seizures that don't get recognized by anybody to more obvious clinical seizures.
The next question is, are you aware of any connections with the mother taking Miralax, which is a double dose, during pregnancy?
Absolutely not. Miralax is mainly, it's something used for constipation, and it largely lives in the gastrointestinal tract. So keep in mind, the gastrointestinal tract is really essentially outside our bodies. I know it sounds weird because it's all inside of us, but it's surrounded by a mucosa that protects it from toxins like poisonous mushrooms and other things. And Miralax is something that is called an osmotic agent. It draws water and takes it through the GI system, which helps push stool out from the rectum and the colon. But it should not affect, as best I'm aware of, anything related to autism or birth defects when taken by a pregnant mother.
Do we know anything about the impact of COVID on kids with autism or kids with special needs?
I don't think, it's been looked at. Obviously, COVID has now killed 1 million people around the world and infected 100 million people. And certainly many individuals with autism, many with epilepsy have been infected with COVID. To the best we know, no, there's no special risk. We don't think they're at greater risk to develop serious complications of COVID, like pneumonia or the cytokine storm from their immune system. And we don't think the same is for epilepsy as well. I mean, yes, if you have epilepsy and you get a fever of 103 degrees from COVID, yeah, you could have a breakthrough seizure, but you could get that from a severe cold or flu as well. So nothing special about COVID for this population.
Is there any connection between autism and a particular type of epilepsy?
No, interestingly, as I said, I think focal epilepsy is most common, but it's most common in people without autism. So in that way, it seems to somewhat mirror the general population. I think individuals with autism may have some may have severe forms of epilepsy, some may have mixed focal and generalized, but overall, individuals with autism pretty much parallel the overall epilepsy community, obviously with higher rates of intellectual disability and behavioral disorders, but as far as the seizure types, pretty similar.
How can we manage febrile seizure episodes even though a child is on epileptic drugs?
So depending on the case in the child, so it's very different. Most children with febrile seizures have simple febrile seizures. They last less than 5 minutes. They stop on their own. And little needs to be done for that child other than observing them. As I said, potentially one of the more dangerous things is if a febrile seizure occurs in the middle of the night, parents don't know what's going on. It's prolonged, which they can be in 10, 15% of cases. And those can cause probably brain damage if the seizure goes on for 30, 45 minutes and can lead to seizures without fever later in life in childhood. And occasionally, very rarely, single seizures in sleep can be deadly. whether they be febrile seizures or afebrile seizures. So I think, nothing specific, for the most children. Certainly, for example, Dravet syndrome is a disorder often presenting with febrile seizures in the first year of life. Those children have very high rates of prolonged seizures. And so if I have a patient with Dravet syndrome and they have a seizure, to be honest, with or without fever, and they've had a history of prolonged seizures, I would give rescue medication like a rectal diazepam or inhalational midazolam as soon as the seizure begins. Just put the fire out before it gets going.
Nikisha, and I just want to remind you that we're just 5 minutes left for Dr. Devinsky to be with us. So we'll take one last question. I know we had several questions, and thank you everyone for putting them down here, but we are really running out of time. So we let Nikisha just take one last question. Thank you.
Thank you, Mariam. The last question is, what are your views on the microbiome-based drugs for ASD?
So I think the microbiome is an exciting area. I think there is absolutely no doubt that it's important in many aspects of health and disease. It probably has to do with why we metabolize some drugs and absorb some drugs better for some people than others. Probably has to do, we know in mice, you can take the microbiome, the stool from an obese mouse and put it into a thin mouse, and the thin mouse will gain weight or vice versa. So the microbiome, is related to many things. There's a gut-brain axis, and some things made by the microbiome may change the permeability of the brain to different compounds in the blood. So the bottom line is it's very complicated. We're at the early stages scientifically of studying the microbiome. And I would say anyone who thinks they know how to improve autism by changing the microbiome is practicing religious belief. It may be based on some anecdotal observations, but that's like, me saying, I know that brain waves are important in mental function, which they are, and I can hold a magnet over your brain and change your brain waves, and I'm going to make you smarter. Holding a big magnet over your head and saying I can change your brain waves a little bit may be true. Saying I can make you smarter would be a lie, in my view. So I think the microbiome is an important thing, and certainly avoiding processed foods is a common sense thing because we know that the microbiome is determined to a large degree by what we eat. Antibiotics are probably the most radical thing to disrupt the microbiome. It just totally changes the species of bacteria living in your gut. So those are probably, and they're not natural antibiotics, they're made in pharmaceutical companies. So I think if you wanted to have a healthy microbiome, as much as you can avoid antibiotics, number one, and number two, as much as you could eat unprocessed foods, which I think are the leading causes of disability and death in most nations in the world.
All right. Thank you for that. We're just out of time. Dr. Devinsky, I know you have to be out in 2 minutes, but I just wanted to say thank you so much for being here with us.
Yes, Dr. Devinsky, thank you very much. We really appreciate you being here.
Yeah.
My pleasure. Thanks again.
We wanted you here. We wanted you here physically, but maybe another time after the COVID.
Sounds good.
Thank you, Dr.
Ask you, Dr. Devinsky, there have been a few questions about whether you take consultations, and if so, then where can people reach out to you?
So the challenge for me is I work at NYU Medical Center, and the policy is I can do consultations with people who I have seen in my office, but I technically can't do it with those who have not come to see me in New York City. I'll find out if there's any policy changes on that, but for now, that's the policy of the medical school. So, but if it does, I'll let you guys know and you can communicate. I'd be happy to help out.
Thank you. Thank you so much for that. Thank you, everyone.
Thank you.
Here, and we have the video up on YouTube, which also we can share with anyone who's interested. So please write into us. Thank you, Dr. Devinsky. It was
Thank you, Dr. Devinsky.
Many thanks. Bye-bye.
Thank you, everybody, and we hope to see you at our next webinar.